Dear students, I am Bharat Kashyap, lecturer biology at GSSS Majhwad district Mandi. Today I am going to teach you sixth chapter of biology, molecular basis of inheritance, and today's topic is human genome project and DNA fingerprinting. Let us begin today's topic, human genome project. We know the sequence of bases in DNA determines the genetic information of a given organism. If two individuals differ, it means their DNA sequences should also have some differences. This project was launched in 1990 to find out the complete DNA sequence of human genome, which becomes possible due to the advancements in field of genetic engineering. The magnitude of requirements for this project can be imagined if we simply define the aims of the project as follows. In human genome, the approximate number of base pairs are 3 into 10 raised power 9. If the cost of sequencing single base pair, it comes out to be $3, then the total estimated cost of 3 into 10 raised power 9 base pairs comes out to be $9 billion. This is a huge amount of money which is required to accomplish the project. So this is known as mega project. Further, if the sequences which we obtained in this project were to be stored in typed form in books, then we need 3,300 books, each having 1,000 pages and each page containing 1,000 letters to store the informations of DNA sequencing of a single human cell. The enormous amount of data expected to be generated requires the high-speed computational devices for data storage, its retrieval, that is, it can be produced when it is required, and its analysis. Thus, a new interdisciplinary field of science has developed. This is called bioinformatics. Now, let us discuss goals of Human Genome Project. It has following goals. First one, to identify all the approximately 20,000 to 25,000 genes in human DNA, to determine the sequences of 3 billion chemical base pairs that are present in human DNA, to store this information in databases, that is a collection of informations from where it can be assessed, managed, and updated whenever we need. Then to improve tools for data analysis. Then transfer related technologies to sectors such as industries. And last but very important, to address ethical, legal, and social issues, also known as ELSI, that may arise from the project. From ethical, we mean relating to the moral principles of the people, legal, that you know, relating to the law, and social issues, we mean the problems influencing the society due to this project. So these were the main goals of Human Genome Project. Now, the Human Genome Project was a 13-year project. It was initiated by the US Department of Energy and National Institute of Health. During the early years of Human Genome Project, the Wellcome Trust of United Kingdom became a major partner, while later on contributions also came from countries like Japan, France, Germany, China, etc. This project was completed in 2003. The goals of Human Genome Project can be elaborated further as the knowledge of effects of DNA variations among individuals can revolutionize the ways to diagnose, treatment, and prevent thousands of human disorders. The study of DNA sequences of other organisms can be applied towards solving challenges in healthcare, agriculture, energy production, and environmental remediation as well. Many model organisms. These are those non-human species which are extensively studied to understand particular biological phenomenon, and they provide insight into working of other organisms such as bacteria, yeast, Caenorhabditis elegans, which is a free-living non-pathogenic nematode, then Drosophila, the fruit fly, and plants like rice and Arabidopsis, which belong to Brassicaceae family and is useful in genetics due to small generation time, its small size, and prolific seed production. They have been sequenced so far. Let us discuss various methodologies which are used for DNA sequencing. The methods involved two major approaches. First approach is expressed sequence tags, that is ESTs. This method is used to identify all the genes that are expressed in the cell as mRNA and produce proteins. The other method is sequence annotation. In this method, it is the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequences and later assigning different regions in the sequence with specific functions. DNA sequencing involves the following steps. DNA is isolated from a cell, broken into random small fragments. These fragments were then cloned in a suitable host using specialized vectors or carriers. The common hosts where fragments cloned were bacteria or yeast. The vectors of bacteria called bacterial artificial chromosome (BAC) and yeasts were called yeast artificial chromosome. Nowadays, the technique of PCR, that is polymerase chain reaction, is used to amplify or making many copies of each DNA fragment so that it can be sequenced easily. Now, The fragments were sequenced using automated DNA sequencers technique that worked on the principle of a method developed by Frederick Sanger. He was the only double Nobel laureate, and he also determined amino acid sequence in proteins. Each fragment is sequenced from two ends. Some of them will be same, others will overlap with some of those sequences. These sequences were then arranged based on some overlapping regions present in them. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences, which are also known as microsatellites. We will study about them in DNA fingerprinting, the next topic. Now let us discuss the various salient features of human genome. The human genome contains 3,164.7 million base pairs. The average gene consists of 3,000 bases, but sizes vary greatly, with the largest known human gene being dystrophin, which produces dystrophin protein in muscles, and it contains 2.4 million bases. The total number of genes is estimated at 30,000. Almost all, that is about 99.9%, nucleotide bases are exactly the same in all the people. The functions are unknown for over 50% of the discovered genes. Less than 2% of the genome codes for proteins. Repeated sequences make up very large portion of the human genome. The repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Chromosome 1 has most genes, 2,968, and Y chromosome has fewest, 231 genes. About 1.4 million locations have been identified where single base DNA differences, also known as SNPs, occur in human genome. This information is certainly going to help to find chromosomal locations for disease-associated sequences and tracing human history. As is clear from the study, that Human Genome Project was a big breakthrough in field of science, and it will completely change the scenario in medical field in near future. Let us discuss some of its applications. It will enable a new approach to biological research. All the genes in a genome can be studied together. In the past, researchers studied one or a few genes at a time. It helps to understand tens of thousands of genes and proteins work together in interconnected networks. It reveals the genes responsible for diseases like cardiovascular ailments, diabetes, Alzheimer's disease, cancer, etc. It helps to diagnose and treat genetic disorders through gene therapy. It will provide information that help to prevent inherited diseases. Now we are going to study a very important technique, DNA fingerprinting, which is also known as DNA profiling. It is a technique applied to identify individuals on the basis of their respective DNA profiles. It is invented by a British geneticist, Sir Alex Jeffreys, in 1984. It is based on the principle we know genome of a person contain about 3 billion base pairs which are arranged in a definite sequence to give an individual his or her identity. The technique of DNA fingerprinting is based on the fact that DNA of an individual is about 99.9% identical to that of other, but there is a difference of 0.1%. This small difference in DNA is used for the technique of DNA fingerprinting. We know human genome is heterogeneous and it has two regions. First one are exons, also called coding sequences, which carry information to synthesize proteins. Others are introns or non-coding sequences. They only accumulate changes. They are made up of 10 to 100 nucleotide pairs. They are responsible for variation among individuals. They can be used as a tool to create genetic fingerprint of an individual. Some of these contain highly repetitive sequences of base pairs and are randomly repeated many times and found at many places throughout the length of DNA. Number of repeats is specific in every individual and do not match with other. Let us discuss this with an example. Say unit ACA is repeated 55 times at one genome locus, whereas 155 or 115 times at other locus, and so on in an individual. Others may have different number of repeats at other loci. These polymorphic genetic loci are called simple tandem repeats or STRs. The identical twins may have same tandem repeats. Now, these tandem repeats make satellite DNA. For example, there is a sequence. You can see the repetitions here. ATTC, GATTC, GATTCG is a sequence where ATCG is repeated 3 times. Such repeated sequences are of following two types. First one are mini satellite. It is a sequence of 15 to 50 base pairs. and repeated tandemly up to 1000 times. They have 30,000 mini satellite loci present in human genome. So this was mini satellite. Now the microsatellite. It is a sequence of less than 10 base pairs and repeated 10 to 100 times. In human genome, there are 2 lakh such loci. These simple tandem repeats of short sequences are called variable number tandem repeats, VNTR. These are inherited from parents and are genetic markers in a personal identity test. So I do hope you have understood this about the tandem repeats. So in this diagram also we are seeing here this microsatellite, and this is the two bases are repeated several times. This we have already discussed. And if there is a single nucleotide polymorphism, this is called SNP. And in this diagram there are 8 repeats, in this 7 repeats, and there are 9 repeats. So this is the tandem repeats, how they make satellite DNA. Let us discuss the various steps which are involved in DNA fingerprinting process. First of all, isolating the DNA from the sample which may contain the strains of blood, semen, hair follicle, etc. Then digesting the DNA with the help of restriction endonuclease enzymes to make smaller fragments. Next will be separating the digested fragments as per the fragment size by the process of electrophoresis. Then transferring or blotting the separated fragments onto synthetic membranes like nylon or nitrocellulose. Then hybridizing the fragments using labeled VNTR probes. And last will be analyzing the hybrid fragments using autoradiography. So these are the various steps involved in DNA fingerprinting. Let us understand these various steps with the help of diagram. First of all, blood sample is taken. Now DNA is extracted from blood cells. Then DNA is cut into fragments by a restriction enzyme. Now these DNA fragments are separated into bands during electrophoresis. This is done on an agarose gel. Now this DNA band pattern in the gel is transferred onto a nylon membrane by a technique known as Southern blotting technique. Then the radioactive DNA probe which is prepared, it contains smaller fragments of DNA with known sequence, and also they are made radioactive. Now the DNA probe binds to specific DNA sequences on the membrane, and now excess DNA probe is washed off, and at this stage the radioactive probe is bound to the DNA pattern on the membrane, and then X-ray film is placed next to the membrane to detect the radioactive pattern. And lastly, the X-ray film is developed to make visible the pattern of bands which is known as a DNA fingerprint. So this is the whole process which you can understand with the help of diagrams. In the end of today's lecture, let us discuss various applications of DNA fingerprinting technique. Look at the picture. It covers various fields like forensic science, parentage testing, medical diagnosis, use in food industry and agriculture. So there are various important uses of DNA fingerprinting. As discussed earlier, the technique of DNA fingerprinting is used for DNA analysis in forensic tests in case of crimes like murder, rape, etc., and disputed paternity tests. DNA fingerprinting is used to determine the frequency of a gene in a population, which gives rise to diversity. In case of a change in gene frequency or genetic drift, DNA fingerprinting can be used to trace the role of this change in evolution. So with this, we come to close of today's lecture. I hope you must have found it interesting. Thanks for watching.